And without further ado, the floor is yours.
Thank you very much, Dr. Antonio. It's an honor always to be able to speak at an event. I hope to get more in-person events. I've been working, quite a lot, as you mentioned. I retired in actually December of 21, and in March and April of 22. $4 million of NIH grants that I was a co-PI on, and $10 million of DoD grants came in. So the Ohio State kind of got me back at work relative to the work in our brain sciences study and our DoD studies. So it's been an active area. And then I'm also working with sport performance at Ohio State, and then I'm emeritus at Connecticut as well. And you can see. So if I go forward here, I'll mention any of the conflicts of interest I have as we go along. I've done a lot of corporate research, as many people in the industry know, as well as DoD and NIH stuff, but I'll try to mention that. I was affected by the recent conference I was at in Finland at the International Conference on Strength Training, which was really an outstanding conference. But one of the things that came about, and I was talking with colleagues like Greg Half at Australia and a number of other individuals, Paul Comfort from England, et cetera. And I'm finding that there's this new construct that if a study is 10 years old, it's no longer important. And I was somewhat concerned with that, because the literature base is many times based by what we've done in the past and how we reinterpret it in the future and how it may impact what we do today. And if you take away the historical base of our field, especially in nutrition and strength conditioning, you basically remove a lot of the dimensionality of it. So I'm just today, in the limited time we have here, I'm not known for a 30-minute talk. So I will try to just make some major points from some prior studies that we've done, and then look at what they mean today in this type of thing. And basically just wanted to emphasize the fact that all that we do is a combination. One striking feature I got when I was listening to one of the top women in menstrual physiology give her presentation, and she said her organization now have come out that we don't want to see any more reviews, systematic reviews, meta-analyses on the menstrual cycle. There are now more reviews on the menstrual cycle than actual research papers. So we got to look at our different areas of research, and we got to ask the question, when we need a systematic review and you end up with 5 studies, why don't you just tell people to read the 5 studies? When we end up with a meta-analysis and have no narrative and the algorithms are such, we really got to clean up our world with regard to how we look at reviews and how we write reviews and focus on independent investigations. Anyway, I'm off my I'm off my soapbox. A lot of the work that we've been doing over the years has been related to this whole construct that Jeff Bullock and I talked about for many years. Is resistance exercise creating the stimulus, and then basically it results in hormonal release of a variety of hormones, and there's interactions with target tissues and receptors and genes, and that translates, over to the genetic machinery to create either a protein positive, protein negative, and ultimately with training, you're going to get this secretion, and it's going to affect your body functions in many ways. I wanted to mention something that's come to light with us with some of the work we're doing now, is that there's a ton of non-genomic hormonal interactions. Most of us are familiar with steroid hormones, which interact genomically with the DNA upregulatory elements themselves, versus the non-genomic methods of interaction with cells and tissues, which are very multiple in nature. And you'll see that in one of my other slides. So one of the things that we see here in this first cartoon here, you can see that you have the only real, genomic interfaces are the steroid hormones. They're the ones that are actually doing with translation to the actual regulatory elements of the DNA. And obviously, that's one of the reasons why, for example, cortisol interferes with binding on some of those regulatory elements, and we see some of the association with catabolism, etc. Well, anyway, one of the first studies I wanted to talk about that we did many years ago And basically what we looked at was really very much of a high carb, 100 grams and a high protein type of supplement that we were going to give to people when they do very high intensity type workouts. And when you talk about that, we're talking about multiple exercises that are driving the blood lactate levels up, typically on average of 13 to 15 millimolar. And this was part of the genres when people were getting into the high intensity type of circuit training and that type of thing. And the thought was we need to replace carbohydrate, we need to address the muscle damage, etc. And this was really one of the studies that we did for Twin Labs on one of their studies, and it was funded by Twin Labs and some of the other work at Penn State. And the design was pretty straightforward. I've always been fortunate to have Jeff Bolick in my team since 1993. With all the work we've done dietetically, it's been very clean and tight. So the study really looked at a within study design, and we found some interesting things. But the unique thing I wanted to bring out about this particular study is the fact that we were one of the first ones to really look at 3 days of hormonal response to the same workout. And with that, we learned a couple of different things. We looked at growth hormone, which is a much more complex story than I can tell you here today. We looked at serum cortisol, and you can see here a very rapid response, differential response of growth hormone and cortisol. And then they regress to their normal control type of response in the next day. And if we look at that, the other thing I wanted to mention, keep in mind, is that Barry Spearing, another former student of mine, did some really nice work to show that cortisol, one of the impacts on cellular type of aspects is the fact that it can obviate down regulatory streams of the mTOR system, etc. That's something we got to keep in mind. There's so much cybernetic aspects going on. We got to keep in mind that just because you get something up front doesn't mean that it's going to be maintained down to the point where you meet the genetic machinery that we talked about. Anyway, if you look at those 3 days, you can see with 100 grams of carbohydrate, dramatic insulin responses, obviously to manage that. And you look at the IGF, we also see a combination of the protein availability as well as the response of insulin that's kind of augmenting it, these type of things. And finally, I know we heard about testosterone. I always say this, I look at all these ads on the TV about testosterone enhancers. The problem is that you want testosterone to go up when you have a stimulus, an exercise stimulus that creates the receptor actions that needed to be to manifest it. And I'll show you something on that a little bit later. But you can see here that type of supplement does change the testosterone levels, and they seem to not go back to normal. The supplement, because of the protein availability for the amino acids, the protein pits probably optimizes some of those aspects of that. And if you look at SHBG, the carrier protein, you can see that that's also maintained even in day 2. So what is the takeaway? Short rest protocols require recovery nutrition. There's a stressor in the first use of any supplement. We used the supplement leading up to it. So it still became a stressor with that first type of protocol. The acute hormone responses are different in many cases in the first day, especially with growth hormone and cortisol, with some of the hormones, but some of the other hormones such as insulin, IGF, and testosterone all elevate each day. So the anabolic hormones probably remain elevated due to the availability of amino acids, which is far more than is needed. According to Dr. Bolick, about 35 grams of protein is all you can handle in one shot anyway. So there's a lot of type of aspects of oxidative aspects of energy that's going on as well. So it's interesting. And then obviously insulin is managing the high carbohydrate load. This is a diagram, a very limited diagram, but what it shows you is this goes back to the genomic and non-genomic. And this is very short. This shows you one genomic transition of the testosterone and dihydrotestosterone, the sex-linked tissues, but it translates itself in. But look at all the other ones are non-genomic, everything from all the secondary aspects. So T and other hormones are doing so many non-genomic interfaces that's what makes sometimes it complexity to understand how it's affecting a cell or a particular tissue. So you look at these type of things. I wanted to point out this one here, and this just showed you here there was a fact that if you look here at the signaling that happens with regard to testosterone concentrations, that's a signaler for the androgen receptors. So we argued for many years about that. We had whole aspects of studies that show that, hey, when you eat food, T actually goes down. Well, it does because you got upregulation of the receptors pulling it out of the circulation. So there's something we know that related to the fact that the volume of exercise and how it all works is you get a stimulus and then you get a stabilization and then you have degradation because you're migrating into the cell, that T to the genetic component, and then you get upregulation. And the timing of that measurement is so important. Usually that's where people either miss it or measure it too soon and miss the whole phenomenon. Well, we did a study again looking at this from Lanza with regard to the use of L-carnitine L-tartrate. And again, Dr. Bullock has done a lot of work in this line of work as we've looked at it over the years. And when you add this, you know that L-carnitine really has a lot to do with the vacuoles at the circulatory aspect. That either if you're low in L-carnitine, if you tartrate at that part, you get a constriction or not a dilation. If you have it, you get a dilation, so you perfuse the muscle with a better capillary function. Well, what we found in this particular study is that augmented with the carb protein drink versus water, you got this actual work of over the 3 weeks of preparing with supplementation, crossover, again, a complex design you can read more about here in our paper. And then the errata did show you get this acute elevation in androgen receptors from this combination of supplement use, again, with regard to the L-carnitine compared to the water and the Ensure alone. So this whole phenomenon actually elevates or augments the whole receptor phenomenon that you get. So if you look at the finals, the L-carnitine L-tartrate supplementation actually upregulates the pre-exercise androgen receptor content in combination with the protein carb aspect. And basically, the resistance exercise increases with the androgen receptor with the placebo because it hits normally. Feeding will lead to more significant rises in the AR content. And then testosterone is also elevated to, which is suppressed during the use of the feeding because it's being pulled more out, the minimal changes in LH. So post-exercise, resistant exercise feeding could increase these type of testosterone uptakes due to the fact that you're elevating the receptor content and basically stimulating the whole phenomenon. This is another study we did, which again, it was supported by Advocare, I'm on the Scientific Medical Advisory Board, so to show that they funded this a while back, and we were very interested in a pre-exercise supplement to stimulate better exercise training responses. And we found that it was very interesting. You have a creatine component in it, you have L-carnitine, you have a whole bunch of betaine, you got caffeine. It's almost like a compilage of different things that could stimulate individuals pre-workout. It's a pre-workout enhancer. I know that over the years, Drew Brees and the New England Orleans Saints used to use this stuff by the bushel baskets back in 2006 when we were doing our work, and it really seems to enhance stuff. And what we found, it did enhance aspects of performance. It did enhance aspects of the free testosterone and the total T. But again, you're working out as well as growth hormone and IGF-1. But I think it's as a pre-workout, it's getting a stimulation. So you look at all the things with caffeine, creatine, and all these factors are leading to a higher stimulation. So looking at rapid fire, improved vertical jump performance with the pre-workout supplement, augmented hormones, you get reductions. One of the things we did was looking at the recovery process. It was reductions in myoglobin creatine kinase, and this suggested that you get an attenuation of actual muscle damage. So Muscle Fuel was an interesting pre-workout type of supplement that's been used anecdotally by many professional athletes. Here is just one aspect I'm going to finish up with today. I wanted to talk a little bit about this here. I'll talk about hypogonadism. I'll leave Dr. Storr, who's more of an expert on than I am, on this, but we've dealt with this a lot. But I got a slide out, out of place. One last thing I wanted to say here is that we also looked at ginseng and was funded by a Korean company, and we found with women and men in mid-age that ginseng proved to be a very extensive type of protocol, but it proved to be it reduced the reductions in cortisol. It helped antioxidant activities and the measurements of that. It actually, the high dose of ginseng actually reduced muscle damage, as measured by creatine kinase, and looked at some of the attenuated type of stressor response on the hypopituitary adrenal axis. That was one of the focuses of this for stress management with workouts. A very, very interesting study. Getting back to hypogonadism very briefly, and then I'll wrap it up quickly. It basically done a lot of work with ultra endurance runners, and when you look at really this whole phenomenon, we found in several studies that ultra endurance athletes, males in this case, many of them are hypogonadal. And you can see that the type of study we did here was with Dr. Bullock in our faster study. You can see the treadmill run is of about 3 hours, 2 to 3 hours, 3 hours rather, 65% recovery, very demanding ultra distance. And they were looking at low carb versus high carb. So you can see this, despite the type of diet you're on, whether it be a low carb or a high carb, you're still seeing these endurance runners that have, essentially low testosterone or hypogonadal type of responses. And if we looked at the Ironman championships with Dave Hooper, we went out with Dr. Marishnak and a whole group out in Hawaii. And you can see that you have a similar response. Many of the subjects are really hypogonadal, as demonstrated by T levels that we collected. Now, whether that's true or not, and you see the same thing in the Western states. When we looked at the Western states, a lot of them are very low relative to their T values. But working on some of the aspects with regard to why this might be, it might be because of caloric insufficiency, similar to what you see with the women's triad. And Dave Hooper has written a lot about this along with Dr. Hackney down at North Carolina. And you can see again, you see this lower response and compared to controls. Now, one of the things you got to realize, Dr. Bassan and I was on this position stand for ACSM, and we know that you just can't take a testosterone measure and say, hey, it's low, you're hypogonadal. That's a mistake, and that's where the increases in testosterone patches and everything else has gone up because of the fact that you need a full clinical profile from morning teas, from looking at clinical symptoms that are associated with it, confirmed with additional tests. And if you look at the new ACSM of a few years ago, our update on it by Dr. Bassan and the team, it goes through extensively how you want to make sure that you basically verify that somebody's truly hypogonadal or exercise-induced hypogonadal, which may be due to the nutritional component and the caloric component as well. And then the other thing is we don't want to make fear men who need this type of therapy go away from it because of the stigma of having a testosterone patch and thinking you don't need it. That's not good either. So with that type of aspects, there's a whole bunch of interfaces with hormones. Thank you very much. I'll take some questions, and this is probably a record for the shortest talk I've ever given in my career.
Thank you, Dr. Kraemer. That was a great presentation. I always enjoy it. I have a question about, you showed data on L-carnitine, L-tartrate increasing, I think it was increasing AR levels, androgen receptor. I haven't seen too much on that since that particular study. What are your thoughts on it in terms of, would it make men and or women more sensitive to androgen?
Well, no, I think what happens, first of all, you got to realize if you carefully look at our design, you had an Ensure product that was given, either had the Ensure with L-carnitine or you had the Ensure with water. And with water, it didn't quite have the impact that the L-carnitine. Now, resistance exercise alone, the protocol we had did that. I think personally, it's nutrition, as many of the experts that are on this panel and have done today been speaking, are well versed in this. Polynutrition is so vital, and the genetics between different people, what we're learning now about brain function in our lab, and also how the differentials between type 1 and type 2 muscle fibers very different from extracellular matrices, etc. I guess my thought on the L-carnitine is that one of the key factors that we did early on was the fact that in the capillary circulation, you have a constrictor of vacuole that, if in fact there's that constrictor that opens up the pool of blood to an area, if it's anaerobic exercise, if it's not high enough, it will in fact close down. It won't open up. So I think when you supplement with L-carnitine, one of the subcellular mechanisms is that it allows for better response to anaerobic exercise, which is very hypoxic, and which, if you don't have enough L-carnitine, the capillary circulation closes down versus opens up. So I think a lot of it has to do with the circulatory aspect that might be promoting more T getting to receptors just because of circulation. We also know in the testes itself, from some of the animal studies we've done, is that you've got a blood flow control of a latch mechanism. Actually, interestingly, it's a beta endorphin mechanism that basically opens up or closes, the blood flow. So blood flow response in the testes also very important relative to allowing for blood translation of hormones to signaling tissues and cells.
One last question before we transition to Dr. Storer. Some of the older data looking at different resistance training protocols and how it impacts acutely, how it impacts GH and testosterone. Could you comment on those, whether or not the acute changes in these hormones, how it's related to the volume and or intensity of the resistance training protocols?
Yeah, I mean, that goes way back to the first symposium we presented back in 1987 with what was a historic symposium at ACSM, the first one, and then published. I think the work that's gone on is that basically we know from that study that with what the hormones are trying to do is support the demands for remodeling, repair, and homeostasis of what those tissues that have been activated are. So if you do a very small amount of volume of tissue, there's not much of a need for the endocrine system to throw out a lot of help. So all the physiological systems are responding to the volume and the amount of recruitment. Size principle plays a major role. The more tissue that's recruited and activated, the more support it needs, and thus more endocrine system gets involved, the immune system gets involved, cardiovascular system. All the systems are measured by the amount of recruitment and the amount of tissue that's involved. Because if I just do arm curls, I got to do a heck of a lot of arm curls to get very much of an endocrine response, because it's a small muscle group and my normal homeostatic levels can take care of it. And we showed that in our 1990 study in men, 1993 in women, and we've done subsequent ones on that. Volume and the amount of intensity and the metabolic challenges all regulate those hormonal changes.